Hello everyone, now we are going to learn about cardiovascular system. So this is going to be a lecture series which talks about cardiovascular system and human cardiovascular physiology. So in this section we will be learning about organization of the cardiovascular system, properties of cardiac muscle, cardiac action potentials, origin and conduction of cardiac impulse, the normal electrocardiogram, clinical applications of the electrocardiogram along with cardiac cycle, cardiac output and venous return. Then some aspects of regulation of cardiac output along with hemodynamics. We will be learning about microcirculation, then the lymphatic system in detail, later vascular distensibility of the venous system, then determinants of arterial blood pressure along with short-term regulation of arterial blood pressure, then long-term regulation of arterial blood pressure, then regional circulation, its overview, then coronary circulation, cerebral circulation, splanchnic circulation, then fetal and neonatal circulation, how that happens, then valvular heart diseases, then cardiac failure, along with that circulatory shock. So these portions are very important when we consider this aspect as something which is related to our own heart. So it's like we are learning about our own cells, our heart. It's very important. So let's start learning. So in this video, particularly we are concentrating on organization of the cardiovascular system. So our learning objectives include describe the characteristics of the different blood vessels that make up the circulatory system, differentiate between the systemic and pulmonary circulations, outline the basic principles of circulatory function. So when we try to learn this topic, there might be some technical terms or jargons which we need to know before learning about this topic, like capacitance vessels, veins that serve as a major reservoir of blood. Then conducting vessels, large elastic arteries that transport blood at high pressure to the tissue. Then exchange vessels, capillaries, the thin wall together with a high cross-sectional area and lowest velocity of blood flow, facilitating exchange of nutrients and other substances. Then resistance vessels, small muscular arterioles that by virtue of the relatively large portion of the muscle in their walls are capable of offering high resistance to the flow of blood. So the function of the circulation is to serve the needs of the body tissues, to transport nutrients to the body tissues, to transport waste products away, to transport hormones from one part of the body to another, and in general to maintain an appropriate environment in all the tissue fluids of the body for survival and optimal function of the cells. The rate of blood flow through many tissues is controlled mainly in response to their need for nutrients. So, in some organs, such as kidneys, the circulation serves additional functions. So, blood flow to the kidney, for example, is far in excess of its metabolic requirement and is related to its excretory function. You know that, which requires that a large volume of blood be filtered each minute. The heart and blood vessels, in turn, are controlled to provide the necessary cardiac output and arterial pressure to cause the needed tissue blood flow. What are the mechanisms for controlling blood volume and blood flow, and how does this process relate to the other functions of the circulation? These are some of the topics and questions that we discuss in this section on circulation. So, physical characteristics of the circulation. The circulation is divided into systemic circulation and pulmonary circulation. So, because the systemic circulation supplies blood flow to all the tissues of the body except the lungs, it is also called the greater circulation or peripheral circulation. It is also clear that heart is in fact two pumps in series. The right heart pumps blood into the pulmonary circulation while the left heart pumps blood into the systemic circulation. So there are many functional parts of the circulation. So before discussing the details of circulatory function, it is important to understand the role of each part of the circulation. The function of the arteries is to transport blood under high pressure to the tissues. For this reason, the arteries have strong vascular walls and blood flows at a high velocity in the arteries. The arterioles are the last small branches of the arterial system. They act as control conducts through which blood is released into the capillaries. Arterioles have strong muscular walls that can close the arterioles completely or can, by relaxing, dilate the vessels several fold, thus having the capability of vastly altering blood flow in each tissue in response to its needs. These blood vessels are sometimes called resistance vessels. So when we try to learn about the distribution of blood in presence or percentage of total blood in the different parts of the circulatory system, pulmonary circulation about 9%, superior vena cava is there, aorta is there, heart about 7%, and inferior vena cava will be there, then systemic vessels, systemic circulation about 84% of percentage of total blood. Then arteries about 13%, arterioles and capillaries 7%, then veins, venules and venous sinuses about 64%. So the function of the capillaries is to exchange fluid, nutrients, electrolytes, hormones and other substances between the blood and the interstitial fluid. They are therefore sometimes called exchange vessels. To serve this role, the capillary walls are thin and have numerous minute capillary pores permeable to water and other small molecular substances. The venules collect blood from the capillaries and gradually coalesce into progressively larger veins. The veins function as conduits for transport of blood from the venules back to the heart. Equally important, they serve as a major reservoir of extra blood and are sometimes referred to as capacitance vessel. Because the present pressure in the venous system is very low, the venous walls are thin. Even so, they are muscular enough to contract or expand and thereby serve as a controllable reservoir for the extra blood, either a small or a large amount depending on the needs of the circulation. So, volumes of blood in the different parts of circulation. We learned earlier that lists the percentage of the total blood volume in major segments of circulation. For instance, about 84% of entire blood volume of the body is in the systemic circulation and 16% is in the heart and lungs. Of the 84% in the systemic circulation, appropriately 64% is in the veins, 13% is in arteries, and 7% is in systemic arterioles and capillaries. So the heart contains 7% of blood and the pulmonary vessels contain 9%. Most surprising is the low blood volume in the capillaries. It is there, however, that the most important function of the circulation occurs diffusion of substances back and forth between the blood and the tissues. This function is discussed later. Then cross-sectional areas and velocities of blood flow. If all the systemic vessels of each type were put side by side, their approximate total cross-sectional area for an average human being would be as follows, like aorta cross-sectional area 2.5 centimeter square, then small arteries 20 centimeter square, then arterioles 40 centimeter square, capillaries 2500 centimeter square, venules 250 centimeter square cross-sectional area, then small veins about 80 centimeter square, then vena cava about 8 centimeter square. Note particularly that the cross-sectional areas of the veins are much larger than those of the arteries, averaging about 4 times those of the corresponding arteries. This difference explains the large blood storage capacity of the venous system in comparison with the arterial system. Because the same volume of blood flow must pass through each segment of the circulation, each minute the velocity of blood flow, that is V, is inversely proportional to vascular cross-sectional area, like V is equal to F by A, where V is equal to velocity of blood flow, but F is equal to volume of blood flow. A is equal to cross-sectional area, vascular cross-sectional area. So V is equal to F by A. Thus, under resting conditions, the velocity averages about 33 centimeter per second in the aorta, but is only 1 by 1000 as rapid in the capillaries, about 0.3 mm per second. However, because the capillaries have a typical length of only 0.3 to 1 mm, the blood remains in the capillaries for only 1 to 3 seconds, which is surprising because all diffusion of nutrient food substances and electrolytes that occur through the capillary walls must be performed in this short time. Then, pressure in the various portions of the circulation. Because the heart pumps blood continuously into the aorta, the mean pressure in the aorta is high, averaging about 100 mmHg. Also, because heart pumping is pulsatile and the arterial pressure alters between a systolic pressure level of 120 mmHg and diastolic pressure level of 80 mmHg. And as the blood flows through the systemic circulation, its mean pressure falls progressively to about 0 mmHg by the time it reaches the termination of the superior and inferior vena cava, where they empty into the right atrium of the heart. The pressure in the systemic capillaries rise from as high as 35 mmHg near the arteriolar ends to as low as 10 mmHg near the venous ends. But their average functional pressure in most vascular beds is about 17 mmHg, a pressure low enough that little of the plasma leaks into the minute pores of the capillary walls, even though nutrients can diffuse easily through the same pores to the outlying tissue cells. The respective pressures in the different parts of the pulmonary circulation is very important. In the pulmonary arteries, the pressure is pulsatile, just as in the aorta, but the pressure is far less. So, pulmonary artery systolic pressure averages about 25 mmHg and diastolic pressure averages about 8 mmHg, with a mean pulmonary arterial pressure of only 16 mmHg. The mean pulmonary capillary pressure averages only 7 mmHg, yet the total blood flow through the lungs each minute is the same as through the systemic circulation. The low pressure of the pulmonary system are in accord with the needs of the lungs, because all that is required is to expose the blood in the pulmonary capillaries to oxygen and other gases in the pulmonary alveoli. Now, some very important ones like basic principles of circulatory function. Although the details of circulatory function are complex, three basic principles underlie all functions of the system. Blood flow to most tissues is controlled according to the tissue need. When tissues are active, they need a greatly increased supply of nutrients and therefore much more blood flow than when at rest, occasionally as much as 20 to 30 times the resting level. Yet the heart normally cannot increase its cardiac output more than 4 to 7 times greater than resting levels. Therefore, it is not possible simply to increase blood flow everywhere in the body when a particular tissue demands increased flow. Instead, the microvessels of each tissue continuously monitor tissue needs, such as the availability of oxygen and other nutrients, and the accumulation of carbon dioxide and other tissue waste products, and thus microvessels in turn act directly on the local blood vessels, dilating or constricting them to control local blood flow precisely to that level required for the tissue activity. Also, nerves control of the circulation from the central nervous system and hormones provide additional help in controlling tissue blood flow. Then, cardiac output is the sum of all the local tissue flows. When blood flows through a tissue, it immediately returns by way of the veins to the heart. The heart responds automatically to its increased inflow of blood by pumping it immediately back into the arteries. Thus, the heart acts as an automation responding to the demands of the tissue. The heart, however, often needs help in the form of special nerve signals to make it pump the required amount of blood flow. Then arterial pressure regulation is generally independent of arterial blood regulation. Arterial blood pressure regulation actually. Pressure regulation is generally independent of either local blood flow control or cardiac output control. The circulatory system is provided with an extensive system for controlling the arterial blood pressure. For instance, if at any time the pressure feels significantly below the normal level of about 100 mmHg within seconds, a barrage of nerve reflexes elicits a series of circulatory changes to raise the pressure back towards normal. The nerve signals especially increase the force of heart pumping, cause contraction of the large venous reservoirs to provide more blood to the heart, and cause constriction of the arterioles and many tissues so that more blood accumulates in large arteries to increase the arterial pressure. Then, over more prolonged periods, hours and days, the kidneys play an important or major role in pressure control, both by secreting pressure controlling hormones and by regulating the blood volume. Next major topic is about properties of cardiac muscle. This is a very relevant topic. We just know about cardiac muscles like You might have seen their structure. We know that they have intercalated discs, interconnecting regions. They are contractile. Autorhythmicity is there. Cardiac muscle is excitable. Some aspects like that. But when we try to learn this cardiac muscle topic, there might be some terms which we don't know what is that. Like some technical terms or jargons related to this topic. So our learning objectives include list the special anatomical and functional characteristics of cardiac muscle, compare the structural and functional differences between cardiac, skeletal and smooth muscles, list the properties of cardiac muscle that ensure its non-fatigability. So let's learn some important terms related to this topic like bathmotropy. Bathmotropy relating to excitability. Positively, bathmotropic agent increases excitability, while a negatively bathmotropic agent reduces excitability. So, negatively, bathmotropic agent reduces excitability. Then, inotropy, inotropy, relating to the force of contraction. Positively, iotropic agent increases the force of contraction, while a negatively iotropic agent reduces the force of contraction. Then, Stirling law of the heart, stating that the force of contraction of the heart is directly proportional to the endostolic volume, the pressure within physiological limits. Then, synesthesia, a collection of cardiac muscle cells joined by intercalated discs that allow the entire collection to contract as a single unit. There are two synesthesia, atrial and ventricular, separated from each other by a fibrous band. So, the heart is composed of three major types of cardiac muscles: atrial muscle, ventricular muscle, specialized excitatory and conductive muscle fibers. The atrial and ventricular types of muscles contract in much the same way as skeletal muscles, except that the duration of contraction is much longer. The specialized excitatory and conductive fibers of the heart, however, contract only feebly because they contain few contractile fibers. Instead, they exhibit either automatic rhythmical electrical discharge in the form of action potentials or a conduction of action potentials through the heart, providing an excitatory system that controls the rhythmical beating of the heart. So, first major topic is the anatomical characteristics of cardiac muscle. Now we are entering into this complexity of this topic. So just try to pay attention. Cardiac muscle fibers are arranged in a lattice work with the fibers dividing, recombining, and then spreading again. Cardiac muscle is striated in the same manner as in skeletal muscle. Further, cardiac muscles has typical myofibrils that contain actin and myosin filaments almost identical to those found in skeletal muscle. These filaments lie side by side and slide during contraction in the same manner as occurs in skeletal muscles. In other ways, however, cardiac muscle is quite different from skeletal muscle, as we shall see, like physiological characteristics of cardiac muscle. Cardiac muscle is excitable. Like all other living cells, cardiac muscle cells are excitable. That is, they have the ability to respond to a stimulus. The response to a stimulus is a change in the membrane potential, and if the stimulus provided is of sufficient intensity, a biological response, that is, contraction, is obtained. Factors that affect the excitability of the heart are transferred to as bathmotropic factors. Factors that affect the excitability of the heart are referred to as bathmotropic factors. These factors that increase excitability are called positively bathmotropic, while those that reduce excitability are called negatively bathmotropic. Then, autorhythmicity. Cardiac muscle is self excitatory, although this property can be modulated by nerves, hormones, and drugs. The ability of the heart to generate its own rhythm is discussed later. So, cardiac muscle is contractile. This is self-explanatory. However, cardiac muscle contraction differs in important ways from skeletal muscle contraction, and these are discussed later. So, inotropy refers to the force of muscle contraction. Factors that increase the force of contraction are called positively inotropic, while those that reduce the force of contraction are called negatively inotropic. Then cardiac muscle conducts electrical impulses. The presence of intercalated discs is especially important in allowing cardiac muscles to conduct electrical impulses. However, the speed with which impulses are conducted in cardiac muscle is relatively slow. The heart has in place a specialized conducting system. Then cardiac muscle is a sensation. The dark areas closing the cardiac muscle are called intercalated discs. We know that. So at each intercalated disc, cell membrane fuse with one another in such a way to form permeable communicating junctions. I prefer to search about this structure of cardiac muscle fibers, so you will always remember that structure because you have put effort to learn about that. And just see where this intercalated disc is located. Therefore, from a functional point of view, ions move with ease in the intracellular fluid so that action potentials travel easily from one cardiac muscle cell to the next, past the intercalated disc. Thus, cardiac muscle is a sensation of many heart muscle cells in which the cardiac cells are so interconnected that when one cell becomes excited, the action potential spreads to all of them. The heart action is composed of two sensations: the atrial sensation, which constitute the walls of the two atria, and the ventricular sensation, which constitute the walls of two ventricles. The atria are separated from the ventricles by fibrous tissue that surround the atrioventricular valvular opening between the atria and ventricles. Normally, potentials are not conducted from the atrial sensation into the ventricular sensation directly through this fibrous tissue. Instead, they are conducted only by way of a specialized conductive system called the AV bundle. This division of the muscle of the heart into two functional sensations allows the atria to contract a short time ahead of ventricular contraction. This allows the blood in this atria to empty into the ventricles before the ventricles begin contracting. This division of the muscle of heart into two functional sensations allows the atria to contract a short time ahead of ventricular contraction. This allows the blood in the atria to empty into the ventricles before the ventricles begin contracting. The all or none law in the heart applies to the entire sensation. When discussing skeletal muscle contraction, we talked about the all or none law. For skeletal muscle, a stimulus that is of threshold or greater than threshold intensity applied to a motor nerve fiber will result in the contraction of all the muscle fibers supplied by the nerve, that is, the entire motor unit. In the case of heart, because cardiac muscle behaves like a sensation, a threshold or greater than threshold stimulus applies to any part of the sensation will result in the contraction of the entire sensation. So the action potential in cardiac muscle is prolonged and has a long refractory period. The action potential recorded in ventricular muscle fiber is very important, and after the initial spike, the membrane remains depolarized for some about 0.2 seconds, exhibiting a plateau followed at the end of the plateau by abrupt depolarization. The process of this plateau in the action potentials causes ventricular contraction to last as much as 15 times as long in cardiac cycle as in skeletal muscle. One of the characteristics of cardiac muscle, like all excitable tissue, is that it is refractory to stimulation during the action potential. Therefore, the refractory period of the heart is the interval of time as shown to the left, during which a normal cardiac impulse cannot re-excite an already existed area of cardiac muscle. The normal refractory period of the ventricle is 0.25 to 0.30 seconds, which is about the duration of prolonged plateau action potential. There is an additional relative refractory period of about 0.05 seconds during which the muscle is more difficult to excite than normal, but nevertheless can be excited by a very strong excitatory signal as demonstrated by the early premature contraction of the second example. So the refractory period of atrial muscle is much shorter than that of the ventricles, about 0.15 seconds for the atria compared with 0.25 to 0.30 seconds for the ventricles. The long refractory period is one of the factors why cardiac muscles does not fatigue. So long refractory period is there. Then cardiac muscle require calcium in the extracellular fluid to contract. As is true for skeletal muscle, when an action potential passes over the cardiac muscle membrane, the action potential spreads to the interior of the cardiac muscle fiber along the membranes of the transverse tubules. The T tubule action potential in turn act on the membranes of the longitudinal sarcoplasmic tubules to cause release of calcium ions into the muscle sarcoplasm from the sarcoplasmic reticulum. In another few thousandths of a second, these calcium ions diffuse into the myofibrils and catalyze the chemical reactions that promote sliding of the actin and myosin filaments along one another, which produces the muscle contraction. Thus far, this mechanism of excitation contraction coupling is the same as for skeletal muscle, but there is a second effect that is quite different. In addition to the calcium ions that are released into the sarcoplasm from the cisternae of the sarcoplasmic reticulum, calcium ions also diffuse into the sarcoplasm from the T tubules themselves at the time of the action potential, which opens voltage dependent calcium channels in the membrane of the T tubule. So, calcium entering the cell then activates calcium release channels, also called ryanodine receptor channels. Ryanodine receptor channels in the sarcoplasmic reticulum membrane, triggering the release of calcium into the sarcoplasm. Calcium ions in the sarcoplasm then interact with troponin to initiate cross bridge formation and contraction by the same basic mechanism. Then, without the calcium from the tubules, the strength of cardiac muscle contraction would be reduced considerably because the sarcoplasmic reticulum of cardiac muscle is less well developed than that of skeletal muscle and does not store enough calcium to provide full contraction. Then the T tubules of cardiac muscle, however, have a diameter 5 times greater than that of skeletal muscle tubules, which means 25 times greater volume. Also, inside the T tubule is a large quantity of mucopolysaccharide that are electronegatively charged and bind an abundant store of calcium ions, keeping them available for diffusion to the interior of the cardiac muscle fiber when the T tubule action potential appears. Then the strength of contraction of cardiac muscle depends to a greater extent on the concentration of calcium ions in the extracellular fluids. In fact, a heart placed in a calcium-free solution will quickly stop beating. So, a heart placed in a calcium-free solution will quickly stop beating. The reason for this response is that the opening of T tubules passed directly through the cardiac muscle cell membrane into the extracellular spaces surrounding the cells, allowing the same extracellular fluid that is in the cardiac muscle interstitium to percolate through the T tubules. So, consequently, the quantity of calcium ions in the T tubule system, like the availability of calcium ions to cause cardiac muscle contraction, depends on a great extent on the extracellular fluid calcium ion concentration. Then cardiac muscle cardiac muscle contraction is depend on the initial length of muscle fiber, frank striling low of the heart, and concepts of preload and afterload. The degree of tension on the muscle when it begins to contract is called the preload, and the load against which the muscle exerts its contractile force is called the afterload. After cardiac contraction, the preload is usually considered to be the end diastolic pressure, and the ventricle has become filled. The greater the end diastolic volume, like ventricular filling, and the greater the corresponding end diastolic pressure, the greater is the stretch of the cardiac muscle. This increased initial length of the cardiac muscle results in a greater force of contraction, within physiological limits and is referred to as frank Starling law of the heart. The relationship of length to contractibility is modified by the presence of inotropic factors. For instance, intense sympathetic stimulation can increase the heart rate in young adult humans from the normal rate of 70 beats per minute up to 180 to 200, and lately even 250 beats per minute. Sympathetic stimulation also increases the force of heart contraction to as much as double the normal value, thereby increasing the volume of blood pumped and increasing the ejection pressure. This sympathetic stimulation often can increase the maximum cardiac output as much as twofold to threefold, in addition to the increased output caused by the Frank Starling mechanism already discussed. So, conversely, inhibition of the sympathetic nerves to the heart can decrease cardiac pumping to a moderate extent in the following way. Like under normal conditions, the sympathetic nerve fibers to the heart discharge continuously at a slow rate that maintains pumping at about 30% above that with no sympathetic stimulation. So, therefore, when the activity of the sympathetic nervous system is depressed below normal, this decreases both heart rate and strength of ventricular muscle contraction, thereby decreasing the level of cardiac pumping as much as 30% below normal. The afterload of the ventricle is the pressure in the aorta leading from the ventricle. The importance of the concepts of preload and afterload is that in many abnormal functional states of heart or circulation, the pressure during filling of the ventricle, the preload, the arterial pressure against which the ventricle must contract, the afterload, or both are altered from normal to a severe degree. So, cardiac muscle does not fatigue. It is truly amazing that the heart continues to beat in a healthy individual with such regularity the entire lifespan of an individual without getting tired. Even the most well trained skeletal muscle and the most elite of athletes cannot achieve this. There are several reasons why cardiac muscles does not get fatigued. Cardiac muscle has a long refractory period. This cardiac muscle cannot be titanized, while tetany allows for the generation of greater force. Fatigue also sets in much faster. So, cardiac muscle has a rich blood supply. In context, cardiac muscle has a capillary density that is about 4 times greater than skeletal muscle. Cardiac muscle undergoes aerobic metabolism. Thus, the accumulation of lactic acid, an important factor in the development of fatigue in anaerobically exercising muscles, plays no role in cardiac muscle. Cardiac muscle is rich in mitochondria. Mitochondria account for almost one-third of the cardiac muscle by weight and are best appreciated by an examination of the histology of cardiac muscle. So, when we start a comparison of skeletal, smooth, and cardiac muscles, when we learn about skeletal muscle, its striated distribution of myofibrils orderly along myofibrils, then T-tubules are present, intercellular connections, no types, slow and fast type, functional features, sarcoplasmic reticulum, source of C ions for activation, sarcoplasmic reticulum, spontaneous electrical activity and contraction, not happening in case of skeletal muscles, but duration of action potential, few milliseconds, response to rapid multiple stimuli, tetanization happens in case of skeletal muscle, but susceptibility to fatigue is very high in case of skeletal muscles. Then regulatory response to nerve stimulation, excitation, contraction method, response to hormones, small type of responses. But when we talk about cardiac muscles, we need to learn some important points. When we just look at the structure, you could see it's striated distribution of myofibrils orderly along myofibrils. Then T-tubules are present, intercellular connections, many intercalated discs are there, but there won't be different types. It is one type of cardiac muscle. Then functions and features include sources of calcium ions for activation, sarcoplasmic reticulum and extracellular fluid. Spontaneous electrical activity and contraction is happening here. So duration of action potential variable from 10 ms spike potential to prolonged potentials with a plateau phase. But when you talk about susceptibility to fatigue, it cannot be detonized, and it's resistant to fatigue. And then regulatory response to nerve stimulation, excitation or inhibition will be there. Then response to humans, large. So with this video, if you paid attention, you learned some aspects of cardiac muscles, its properties, importance, some major points, along with some important points related to organization of the cardiovascular system, like functional parts of the circulation, and some glossary terms or jargons associated with this cardiovascular physiology. So with the help of next video, talking we will be covering portions related to cardiac action potentials and some topics related to origin and conduction of the cardiac impulses. So thank you for listening.